Our lecture today. Before we get started with the introduction, I just want to remind everyone that we have a happy hour, a virtual happy hour this afternoon at 5. You should have all received the Zoom invite already. If you haven't, please shoot me an email and I'll send it to you. But hopefully you can join us. It is, of course, BYOB or whatever else you want to drink. Anything is fine. Yeah, hope you can join us. But anyway, today we are very happy to have Jeanine Ash here for our lecture this Friday. Jeanine is a research scientist at Rice University. She got her PhD and her MS in geochemistry from UCLA and a BS in Earth System Geoscience from the University of Arizona. Jeanine is really active in IODP, scientific ocean drilling. She sailed to the Ross Sea on IODP Expedition 374. She sailed to the Guaymas Basin on IODP Expedition 385. And also recently, this February, she was the lead organizer of the IODP Early Career Workshop at Lamont, along with Steve Phillips and me. In fact, that was the last trip that I took before nobody took any more trips again. So, which feels like way longer than 8 months ago. But anyway, Jeanine has really broad interests overlapping chemistry, geology, astrobiology. And today she's going to be talking about the enzyme-level reactions that make and break methane molecules. So with that, Jeanine, very happy to have you here. Take it away.
Thank you so much. Let me get my screen sharing started. That was also the last trip that I took as well, and it does seem like a terribly long time ago. So everyone can see my screen and hear me okay. If not, speak up. I'm just down the road from you at Rice. I would love to visit you in person sometime and eat tacos together. But we'll deal with that when the time comes. This is just a kind of an abbreviated list down here of the number of people, that I've worked with. Before I start talking about science, I wanted to talk about a land acknowledgement. So, as earth scientists, we feel really tied to the land, whether we're doing, field work or the place where we call home. But I think it's really important to remember, particularly as just this past Monday was Indigenous Peoples Day, that the land that we work in, no matter how tied we feel to it, had a long cultural history before us. So where I'm speaking to you today from in Houston, Texas, about 5 different Indigenous peoples sort of overlapped here through the centuries. Unfortunately, many of whom didn't survive, the forced removal from this land and the genocide that accompanied the settlement of this place. And I don't say that to sort of like bring us into a dark place, but because I really feel that, as earth scientists, we're uniquely positioned because we already have these like strong, geological knowledge of the places we are. We could also keep that cultural history with us as we move forward to our work towards a juster and sustainable society. Just shout out to the app that I use here. It's called Native Land. It's a great free resource if you want to understand who was here before you were. So with that, I'm going to start talking about the science part of things. So I'm a huge fan of the X-Files. You know this if you follow me on Twitter. And so I always think about the phrase, "we are not alone." And I'm showing you here pictures of Mars, Titan, and Enceladus. And, when I say we are not alone, it's not necessarily because I believe that we'll find other life besides ourselves in the solar system or even the universe, but we're not alone in many other ways, right? Earth is not the only rocky body. We are not the only body with an atmosphere. We're not the only body that has cool gases that we associate here on Earth with biogeochemistry in that atmosphere, gases like methane or carbon dioxide, molecular oxygen, nitrogen, and hydrogen. But we also may not be the only ocean world, right? So beneath this shell of Enceladus, there is perhaps, a briny ocean, and there's probably liquid lakes of ethane and propane on Titan, okay? So we are not alone in many regards, but in order to really understand the processes that are happening on these other bodies, we need to have a very complete understanding of what's happening on Earth if we have really any hope of interpreting these. In decades to come, we have new missions to Mars that are already on their way there. There's sorry, I said this was Enceladus. Apologies, it's Europa. There's going to be a Europa lander that's sort of in the works. And then, of course, we are sending a helicopter to Titan to fly around. All right, so we're going to be getting more and more information from these things. So I really see my role as a biogeochemist of understanding this one, you know, where we have lots of access to samples and field sites. We can go and we can measure all of the things so that we can hopefully interpret these with some level of realism when we start getting information from them. So, you know, traditionally, the time before COVID, a lot of seminars began with coffee and cookies, right? It's really nice collegial conditions, the sort of, you know, collegial thing we would all do to one another. So if you kind of go there in your mind, that before time when we would all be enjoying coffee and cookies before a seminar, you know, we will have the opportunity to reinvent this tradition when we all, you know, return to working full time at our workplaces. So if you'll humor me, I'm going to talk about 3 potential ways we could sort of reinvent this tradition in a long metaphor. So we could do a free-for-all, like these kids, you know, running amok, hunting for Easter eggs. I've been to one of these with my niece. It's a lot like this. Everyone is just rushing around, taking everything that they can get, no trading, okay? So in this coffee and cookies scenario, you know, you would all rush to the table, you would take whatever was nearby and just hoard it like a dragon. So that would be one way we could reimagine coffee and cookies. We could try to introduce some fairness into this process, sort of a random distribution of coffee and cookies. We could all get to the table, roll the dice. You know, odds, you're getting decaf, evens, you can get, you know, caffeinated coffee. And, you know, the amount of, you know, decaf versus regular coffee that we would have would sort of play a role in, you know, how much of those things we could distribute in this fair process. And then finally, the third potential scenario is sort of like a UN style negotiation. So we all show up, and before anyone walks away with anything, we sort of, you know, go back and forth and say, you know, oh, Chris, I see you've got an oatmeal raisin cookie. That's actually my favorite. I'd be happy to trade you this Oreo. You know, and at the end of the thing, I might have, you know, like half an Oreo, half a chocolate chip cookie, and I might have like cream and sugar for my decaf. It's not my ideal, but it's you know, it's better than maybe either of these other 2 scenarios. All right, and you can imagine that these, you know, 3 different coffee and cookie distribution processes would leave, you know, fingerprints on our departments, right? So it might be like a lot of complaints to the ombudsman in the free-for-all situation. We'd probably be like neutrally indifferent about the random distribution, and a department that implemented this negotiation style, that could be like a really collegial place to work, you know, a lot of collaborations. You would know who your trade buddy was, you know, so different places. And of course, you know, I'm not really talking about coffee and cookies. This is a metaphor. I'm going to be talking about chemical reactions, and chemical reactions also leave fingerprints. And these different styles of coffee and cookie distribution, you know, represent the different types of chemical reactions we could have, such as kinetic, which is similar to the free-for-all. This is, you know, a reaction that runs forward really quickly with no real time to think about how those resources are being arranged in the product. Or we could have, you know, the equilibrium reaction, which is similar to negotiation. So we're going to take our products, we're going to sort of rearrange them around, you know, based on our environmental conditions until we've sort of reached a nice status that we all like to live with. And then this middle one here, stochastic. This is, of course, not a chemical reaction, but this is just basically like the statistical distribution of things. So given the products that I have, how could they, you know, potentially randomly be arranged into products? Given the reactants I have, how could we rearrange them into products, okay? And this ends up being just a really convenient way to think about how kinetic or how in equilibrium something is. So we're sort of going to use this as a foil to these other 2. All right, so how are we going to trace these reactions? I'm an isotope geochemist, and using isotopes to trace chemical reactions is something, you know, that's been done literally, you know, since we've been measuring them, you know, starting almost a century ago. And so you're probably all familiar with stable isotopes, just these single rare isotope substitutions into molecules. So here we have a 13C in a molecule of methane, or we have deuterium in a molecule of methane. And we measure these and compare them to some standard and report these little delta values, okay? So what do these stable isotopes actually trace, right? We have a substrate or a pool of reactants. They undergo a process, a chemical reaction, and we end up with a product. And stable isotopes are going to tell us something both about the substrate and the process, right? Many of you might be familiar with delta ATNO or magnesium calcium as, paleo oceanography proxies, but we all know that when we look at the oxygen isotope composition or the trace metal composition of a carbonate, we're looking at something that's got two pieces of information in it. It's going to tell us both about the temperature that equilibrium carbonate was formed in, but it's also going to tell us about the seawater. What was the oxygen isotope composition of the seawater, or what was the magnesium or the salinity of that seawater, all right? So when we interpret that proxy, we have to sort of keep in mind that it's got two pieces of information in it, and we have to somehow figure out how to pull them apart together. So since I'm mostly going to be talking about methane, so here's an example of how isotopes of methane are used as a fingerprint. So we've got our carbon isotopes down here on the x-axis, and then hydrogen isotopes on the y-axis. And all these little dots here are measurements that have been made through time. This is a big compilation. All right, and so you can see, there's different ways of making methane. Microbes can make it. It can be made thermogenically through the breakdown of organic matter. And then there's like a whole host of abiotic reactions that are, going to take rocks and water, molecular hydrogen, and they're also going to turn it into methane. All right, so let's make sense of this diagram. So biology is fundamentally lazy. It does not want to work hard for what it has to do because that takes energy, and we'd much rather spend that energy, like building bodies, okay? And so they're going to use the lightest isotopes of carbon they can to make that methane. So that's sort of why we have the microbial all the way, in this very negative part of the diagram. So thermogenesis, breakdown of organic matter, well, they're breaking down, organic matter that maybe is made of the bodies of the microbes that made this methane. And so it's going to be a little bit heavier. And then we have a pretty broad range in the abiotic in terms of the carbon sources that they can use, because that's, any rock that contains carbon on Earth could potentially be a source for this abiotic methane, all the way from the mantle to something closer to the surface that's like a carbonate. All right, and then the hydrogen isotopes are even more confusing. You can see there's like, almost 200 per mil spread in this, and that's because a lot of the hydrogen that ends up in methane is coming from water. And so there's a huge range of hydrogen isotope variation in water on Earth. So you can start to see some problems with this, right? We have, all these different substrates, as well as these three different pathways. That's our substrate and our process that's going into the signal. And so this leads to there being a lot of overlap in this diagram, right? It's really hard to say, well, where is the boundary between microbial and thermogenic, or even thermogenic and abiotic? And then once we make that methane, it can change because there's all these different processes, physical process like diffusion, or oxidation. So for instance, microbial oxidation is one I'm going to spend some time talking about today. Again, remember, biology is lazy, so microbes are going to eat the lightest methane, leave the heaviest stuff behind. So we could start with something that was made here, maybe by methylotrophy, maybe by abiotic methane. Microbes could eat it, and then they're going to leave behind this pool of methane that's getting heavier and heavier, and sort of dragging us into this region that's maybe abiotic, maybe thermogenic, hard to say. Okay, so definitely lots of great information here. A lot of what we know about the methane cycle comes from this type of analysis, but it's not perfect. All right, so to get around that, I use molecules with more than one heavy isotope to trace these reactions. These are called clumped isotopes commonly. And so you can see same methane molecules here, except now each one of these has two rare isotopes in it, a deuterium and a carbon for this one, 13CH3D, and then two deuteriums here for 12CH2D2. And we report these as big delta values to distinguish them from the little delta values. And a key difference here is we're not comparing this to any kind of standard material. Instead, we're comparing it to that stochastic distribution, okay? So just what if we took the bulk isotopes we had, we randomly put them together, how many of these molecules would we have here? And what this allows us to do is, again, we have the substrate undergoing a process, we end up with a product. That allows us to basically use this information just to infer things about the process, okay? And we can kind of ignore whatever our starting material was, because we're really interested in the chemistry that's putting 2 of these rare isotopes on a molecule at a time. All right, and so broadly, again, our 2 processes are going to be kinetic reactions or equilibrium reactions, and we're going to compare each of these type of reactions to that stochastic abundance to try to figure out, well, how kinetic or how close to equilibrium is it? All right, so you're going to see a lot of these plots. So this is how I think about this. So if we look at a double isotopologue plot, we've got 13 CH3D on the bottom, 12 CH2D2 here on the y-axis. This black line here with temperatures on it represents thermodynamic equilibrium. So any methane molecule that is formed at thermodynamic equilibrium is going to be on this line. All right, ranging from 0 degrees up to 1,000 degrees. All right, anything that's off that line, there's other information there, and we're going to talk about all the different information there can be. But there's lots and lots of processes, both physical and chemical, that can get you off of this line. So for instance, we could be mixing between 2 different pools of methane. All right, and that's going to give us these curves. This one is very exaggerated, but they could be in any direction, above or below the line, at a number of sort of different angles. We could diffuse a gas, right? You could imagine a gas that's traveling through the subsurface. Nothing is happening to it chemically, but it's moving from one place to another, and maybe the pore spaces are very small. So our diffused gas will sort of evolve in this direction, while the residue left behind would evolve in the other direction from this starting point here. Kinetic reactions are going to go very quickly, and I'll talk more about this later, but as these reactions go fast, it gets harder and harder to put more of these heavy isotopes on there, so we can get these very depleted signals. You can see we're down here in negative 12 CH2D2 or negative 13 CH3D. And we could also oxidize, and I'm showing you here aerobic oxidation. So this is oxidation that might happen, in the upper layers of soil or in the atmosphere, where we're removing methane basically with some sort of oxygen metabolism or chemical reaction. Okay, and I'm going to spend the rest of the talk going over this plot in detail, but this is where we're headed. Okay, so what we would like to do is just to take a diagram like this, do a bunch of experiments in the lab, take a bunch of samples from environments where we think we know something about the methane that's there, and populate a diagram like this and see if different processes sort of end up in distinguishable regions from one another, or are we going to have the same problem that we have with the stable isotope plot and just have a bunch of stuff that overlaps and it's really hard to tell what's going on. Okay, so that's kind of where we're headed. So I'm going to talk just 2 slides about the methods, and then we'll get right into it. People see these molecules written out, 12 CH2D2, 13 CH3D. They look really weird. You're not used to seeing so many isotope substitutions. So I often get asked, oh, are you labeling? Is this some sort of labeling thing? It's not. We measure these at natural abundance, but they are very rare. Another metaphor, if methane molecules were people, which honestly is something I think about a lot because methane molecules are just a lot quieter than people, then the total methane population on Earth would be 7.5 billion, right? It'd be 7.5 billion methane people on Earth. About 83 million of those, equivalent to the population of Germany, would have just a rare carbon substitution. So we'd have 83 million 13C H4s. 4.6 million of them, equivalent to the population of Alexandria, Egypt, would just have 1 deuterium substitution. So we'd have a 4.6 million 12 CH3D. All right, getting into our doubly substituted molecules, things drop way down. Only 52,000 of the 7.5 billion people on Earth, equivalent to Montrose, Texas, a neighborhood that's near Rice, would be 13 CH3D. And then finally, our rarest molecule, 12CH2D2. This would be like looking down at the 7.5 billion people on Earth from a satellite and pulling out just the 1,000 people that annually overwinter in Antarctica. Okay, so very, very rare. So how do we measure them? We have a really big tool. So this is the new instruments panorama housed at UCLA. It was the prototype for instruments like this. There are only 3 that exist right now, and there's 2 more in production. And it's the world's largest gas source isotope ratio mass spectrometer. So we take purified gases. They end up here. We put them on this end. This is the source. There's a hot metal wire in here that ionizes these molecules. They go rushing through this huge electronic sector. There's an enormous 3.5 ton magnet, these orange bars that you're seeing behind it here. That's actually like an engine block lift that had to be assembled in the room. But once it was assembled in the room to lift the magnet in place, there was no way to get it out of the room. So there's just permanently an engine block lift that sits in this lab. And then so the magnet, helps spread these molecules out, distinguished by their mass. And they end up here in the collector. So there's slits that collect the 3 most abundant isotopologues we look at. And then this long tube down here is an ion collector that helps us quantify the low abundance, the 12CH2D2, ranging from 10s to 100s of counts per second. Okay, and we don't just use this for methane. There's a number of other gases here. And then this is about how much analyte we need in order to make these types of measurements. Okay, so on to data. So the first thing I want to talk about is how methane molecules are made, right? If we're going to start talking about what kind of methane do we have here, we need to understand its origin, its source. And there are 3 main pathways for this that each have subgroups: abiotic, thermogenic, and microbial. I'm going to talk about abiotic and thermogenic methane just incredibly briefly, and spend the most of our time talking about microbial methane. So there are many different pathways for abiotic methane formation, but you can think of them sort of on sort of like a sliding scale, from things that happen when it's very, very hot, like as magmas are cooling, all the way down to very low Earth surface temperature type reactions. So some of these hot ones might be silane decomposition. So these are experiments that were done at Carnegie. And you can see as the temperature is hot, we get something, that looks pretty close to thermodynamic equilibrium. All right, so these are about 600 degrees. But as the temperature of these experiments decreases down to 300, and then moving to sort of a different type of abiotic methane formation, the Sabatier reaction. So this is a low temperature sort of water rock interaction that you might see like in the serpentinizing system. We get very, very negative in 12CH2D2, okay? So these are very close to equilibrium if we just looked at the 13CH3D, but they definitely don't have anything to do with temperature at all when you look at them on the 12CH2D2 axis. All right, and one hypothesis for this type of pattern is hydrogen tunneling. Essentially, it takes a lot of energy to sort of like overcome to get 2 deuteriums on a molecule. And so as you lower the temperature, this becomes less and less likely. And so you get these huge depletions because you're much more likely just to make 12CH3D or even just a 12CH4 than you are a 12CH2D2. Okay, and so this is sort of one hypothesis for why we see this relationship with temperature and the different abiotic methane formation mechanisms. Okay, so now we know where abiotic methane lands on this plot. What about thermogenic methane, right? This is a very important, natural energy resource, and a lot of people are interested in this. And so here are 2, they're hard to see because they're overlapping very tightly. What we think of as classic thermogenic methanes, one from the Marcellus and another from the Utica shales. They're basically in thermodynamic equilibrium. And the temperature, although it's not labeled here, is about 160 degrees Celsius. So that's right basically smack in the middle of the gas window, somewhere between 100 and 250 degrees. Okay, so there's more to the story, but for today, sort of the classic case scenario is thermogenic methane is going to be on this thermodynamic equilibrium line, somewhere in the gas window temperature. All right, and so microbial methane formation, this is my baby. All right, so whenever I think about microbes or any sort of, biogeochemical gases that's getting made, I'm always thinking about the enzyme that's actually doing the work. Okay, so this is the enzyme that we're going to talk about the most today, methyl coenzyme M reductase, or MCR. All right, it's this big, gnarly dude here, and all these bright spots you see in the middle, these are like little metal centers, nickel, manganese. These are like hot spots for catalysis, right? So just like we're taking in oxygen right now, we're metabolizing it, using iron in our blood, and then spitting out CO2. They're taking in their food, it's being catalyzed on an enzyme, and then they're spitting out methane, okay? So there are three sort of classic divisions in microbial methane formation in terms of metabolisms. There's acetoclastic methanogenesis, which I'm actually not going to talk about today, so no reaction here. But the two I am going to spend a bit of time on today are hydrogenotrophy. So this is taking a metabolism that takes CO2 and molecular hydrogen, spits out water and methane. And then methylotrophy, which takes a methyl group, could be any number of different types of methyl groups, but breaks it down, spits out CO2, water, and then also methane. Okay, so the landscape of methanogenesis is constantly changing. So even within the last decade, there have been two new metabolisms that represent, whole new branches on the methanogen tree of life that I'm going to talk a little bit about today. And that the first one here is methoxidotrophy. So we often find, methane biogas associated with coal deposits. Part of that is just the process of making coal. But there are also microbes there that eat these really nasty, complex coal molecules, that would give you and me cancer, but they're loving it and they're making methane out of it, okay? And that metabolism is called methoxidotrophy. All right, we've really only known about that for about the last 7 years. Okay, and then this other one here, I love this guy, Methanomycelicoccus. I had to work so hard to figure out how to say that. All right, this is, I actually need to update this because the community has decided that we're gonna call this hydrogen-dependent methylotrophy. So they're also taking and breaking down methyl groups, but they are obligately dependent on having molecular hydrogen. All right, and the other thing I love about this guy, not only is he commonly found in brackish wetlands and marine settings, lakes, ruminant stomachs, guess where else he is? He is inside all of us right now. So we have Methanomycelicoccus in our guts, and I think it's really cool that we know that we have an archaea partner, right? Because all of these guys, they're not bacteria, they're archaea. So this is our archaea buddy making methane for us. And you would know if you didn't have him because I think it's called trimethylamania or something like that. Not that kind of doctor, but there's actually a disorder you have where, if you don't have this guy breaking down your trimethylene, that's a really smelly molecule and you'll smell like fish. So this is, serious medical condition, not making light of it, but you would know if you did not have Methanomycelicoccus or something doing the job of Methanomycelicoccus. Okay, so, we're trying to figure out what controls isotope pairings, right? So we could make some predictions. We could say, well, abiotic methane made at high temperatures looks like high temperature thermodynamic equilibrium. And then it gets weird when the temperature gets low, but there's potentially this hydrogen tunnel reason for that. Thermogenic methane looks like, gas window temperatures. That's great. So we might predict, okay, well, methanogenesis is happening, even lower Earth surface temperatures. So maybe it's going to be in that low temperature spot. But before we get there, methanogens are complicated. And so, they have all these different metabolisms. Even within the same metabolism, you can switch them, from one methanol group to a different methanol group. And so what's going to control their distribution on that double isotopologue plot? And so I sort of designed some experiments with collaborators at Radboud University, Stephanie Berger, Mike Yetten, and Cornelia Velt, to figure out, what's going to drive that. Is it going to be the metabolism? Will the metabolic pathways be distinguishable? Is it the organism? So will Methanosarcina mazei always produce the same isotope pairings, even if I'm giving her different food? Could it be temperature, right? If these metabolisms are equilibrating, perhaps in the way that, thermogenic methane is equilibrating, then I would expect to see a relationship with temperature. Or is it the rate of reaction? So if these metabolisms range from kinetic to equilibrating, then I could just expect to see a relationship with the rate of methanogenesis. Okay, so remember, we're thinking originally, oh, this is the temperature that methanogenesis is happening at. They're all going to be up here. Guess what? They are nowhere near that part of the map. Okay, they're down here in this sort of parenthetical swoop that we call lovingly call a microbial array. All right, and so these are all the metabolisms mixed. So right away we could say this has nothing to do with temperature. Okay, so this is not going to be an equilibrium reaction. That's not terribly surprising. A lot of enzymatic reactions are kinetic, right? Because we want a reaction to move forward so that we get that energy out of it right away and can move forward with the next step of our metabolism, whether we're building a body or just trying to survive. Okay, so the next thing I want to do is sort of look for patterns in this. And I'm going to take away all the methoxidotrophy and the methanomycelicoccus style, because we actually don't fully understand the biochemistry behind those metabolisms yet. So I just want to deal with what we know very well about. Okay, so when I take those away, you can see there's sort of two groups here. We have this light blue group that's near, but not quite at equilibrium. It's quite a bit off. And that turns out to be all the hydrogenotrophic methanogens. They group up here. And then this group down here, this warm colored group, that happens to be all of the methylotrophic methanogenesis made methane. Okay, so this is interesting. They're both off the equilibrium line. Hydrogenotrophic methanogens make methane that's a little bit closer, but not quite there. And they're definitely distinguishable from each other. All right, so why? Why are they in disequilibrium? And then why does methylotrophy produce more disequilibrated methane? And to do that, we're actually going to look at sort of the enzymatic process, the metabolisms that make both of these different types of methane. So we'll start over here on the left with hydrogenotrophic methanogenesis. You see we start with our CO2 molecule down here. Now, I am not a biochemist, so forgive me, but the way I think about this is, we start with our CO2, there's, some machinery, and, we're going to add, a hydrogen sort of step by step. So each one of these, progressive steps, you can see by up here, we have a CH2, by up here, we have a CH3, and then we get to our last step, and where I have this yellow star right here, this is our magic enzyme, MCR, okay? And this is where we add the final hydrogen. Now, based on experiments done, for instance, by Dave Valentine at UC Santa Barbara, as well as some modeling, it's thought that some or all of these initial steps are semi-reversible. So semi-reversible to an isotope geochemist means equilibrating, okay? So we could be, moving these hydrogens back and forth, thinking about it each time, and by the time we end up right here, we have a CH3 that's basically in thermodynamic equilibrium. But when we get to this final step with MCR, we're gonna slap a hydrogen on that thing and push it out of our cell because it's waste. There's no sort of energy that can be gained by hanging on to that methane and letting it fully equilibrate with its environment. We want to get it out. And that last step that's not reversible, that basically takes this molecule CH3, with some other biochemistry stuff that was more or less in thermodynamic equilibrium. But when we slap that last hydrogen on, it's no longer in equilibrium. It's out in the world, okay? On the other hand, we have methylotrophy. Okay, so there's a lot of similarities, right? You see all the same, letters here, but just with different machinery, except these arrows are all running backwards. And this is because methylotrophs sort of inherit their methyl group from the environment, right? They're starting with the CH3 that's already formed, okay? And that CH3 goes in right before this MCR step, slap a hydrogen on it, push it out the door, okay? So not only is this step not reversible, but we're starting with just this random molecule that we got from the environment. It didn't spend any time down here with hydrogens moving back and forth isotopically. So there's no equilibration that happens here at all. So what it looks like to me is that the final hydrogen addition of methanogenesis is non-reversible. This causes the disequilibrium in clumped isotopes that we observed in these cultures. And then this also could explain why methylotrophic methanogens produce methane that is even more in disequilibrium, because they don't have the opportunity to sort of reorder any of their methyl molecule before that final step. Okay, what happens if I have the same organism, but I'm going to give them a different food source. So, you know, you got to be versatile to be a methanogen. You're living in some weird places. You're going to take what you can get. Maybe one day it's a methanol, maybe another day it's a trimethylamine. So what happens here? So this one up here, this is Methanosarcina mazii, grown on trimethylamine. So that ends up here. Whereas if I just give it methanol, it ends up down here. Okay, so these are different. This is our Methanomycelicoccus luminensis, growing with molecular hydrogen and trimethylamine up here. But then if I just give it MeOH plus hydrogen, it ends up down here. And then this is one of our coal eaters, our next orthotrophs, Methanococcus shangliensis on TMB. This is one of these weird coal molecules, but it can also just metabolize methanol. So if I give it methanol, that ends up over here. So this is interesting because this seems to imply that if I give the same organisms different food, we're going to get a different clumped isotope composition. So what we need to ask is, well, so is this pathway dependent or is this substrate dependent? So these are the methanogens that grew really well for me, but unfortunately, we don't know a whole lot about their biochemical pathway. So I had to try to answer this in a roundabout way. Because I'm not a structural biologist, I'm not going to be able to image all of these different, biochemical molecules through different parts of this process. If it is substrate dependent, then we would be able to monitor this because we can monitor the stable isotope composition as it changes. So I performed these batch incubations in replicate and was able to stop them at different times of the process. So these are closed system incubations. So I've got my 250 ml vials, they're sealed off. They start with everything a methanogen can dream of, this juicy media, lots of this, food, whether it's methanol or, TMA or whatever it is they're eating. So this is Methanococcus shangliensis grown on methanol. And then as they're eating through this closed system, they have to use the heavier and heavier methanol that gets left behind. Like at first, they're just eating the light stuff. Then they don't really have much of a choice. And they have to, if they want to eat, they have to eat what's left. And so this is the delta 13C and the delta deuterium of the methane that they're producing, okay? And that's representative of the substrate that they're eating, okay? So that substrate is changing. What's not changing at any point in time, though, is the clumped isotope composition. Okay, so, the reason we have to do this way is because I can't measure the clumped isotope composition yet of substances like methanol or TMB. We just don't have a good way to get those into a gas form and measure them, okay? That would be like a whole other wing into this field, one day. So we sort of have to look at, other indicators of yes, the substrate is changing, but does that affect the clumped isotope composition? And the answer is no. Okay, so not a nail in the coffin, but I think this definitely lends evidence to the hypothesis that it's pathway dependent. Just because they're, we think that they're using the same metabolism for these different, the exact same metabolism for these different foods, they might be having to treat these molecules, slightly differently. And you could imagine if someone put down, a glass of methanol in front of you and a piece of coal on the other hand, you don't have to deal with those differently. Definitely work still to be done here, but that's where my thinking is at this moment in time. Okay, and then what if I gave the same organisms the same food, but I made them perform methanogenesis at different rates, okay, to sort of try to figure out, well, can we move things on this diagram by altering the rate of methanogenesis? And I did this by, I created like a superclass of methanogens, like the millionaires. They had all the media and vitamins and food in the world that they wanted. And then for a portion of that population, their global economy crashed and they only had 1/5 of the resources. So they had a diluted media to work in. They were pretty unhappy. They were happy enough, however, to make methane. They just did it more slowly than the methanogen millionaires. And you can see here, this guy, luminensis on TMA and H2. These look the same. So here is our happy guy, unhappy guy. M. shangliensis on TMB. These are overlapping pretty closely. Luminensis on methanol and H2. also overlapping very closely, and Shangliensis on methanol only, also overlapping very closely. So the isotopologue distribution seems to be insensitive to the reaction rate. So the starved methanogens basically produce the same isotopologue distribution as the methanogen millionaires. Okay, now caveats, because I had to do these in the lab. And I was only able to visit at Radboud University for a little over a month. So I had to leave with enough methane to measure. So does that mean, are we at all close to approaching what might be like, you know, subsurface deep biosphere reaction rates? No, those are probably much slower than what I was able to create in the lab. But I was able to get sort of, you know, like almost a factor of 3. Okay, so here are our slowest guys doing about 20 nanomoles of methane per minute, you know, and then our fastest guys doing maybe like 55 nanomoles of methane per minute. Okay, so it is a factor of 2, and again, we see no difference in the clumped isotope composition between these. All right, so if I go back to sort of these original questions about what controls isotope pairings, I would have to hone in on metabolism, you know, based on what I know so far. So it looks like these metabolic pathways, there is a potential that they could be distinguishable from one another. Obviously, with lots and lots of work to be done, I couldn't find any evidence that if we give the same organism different food, it would look different. It's clearly not related with temperature. And within the range of reaction rate I was able to produce, there's also no alteration to the clumped isotope composition. So metabolism, maybe. All right, so imagine my surprise when I try to take this, you know, to the seafloor to figure out how methane is made, because you see this pink group up here. This is unambiguously microbial methane from below the seafloor in the Bornholm Basin in the Baltic Sea. And it does not look at all like the methanogens, methane that was grown in the lab. So why? And that's what I'm going to spend the rest of the time talking about. Because as it turns out, it's not just how methane molecules are made, but also how they're broken and consumed that I think is going to turn out to be a really important part of the story. So I'm going to talk about the anaerobic oxidation of methane, or AOM, and this is a fascinating reaction. Okay, so these methanotrophs are archaea, okay, just like the methanogens. However, they have to perform their metabolism with a bacterial partner. So like, just imagine if you couldn't breathe without like a fungus hanging out with you. Like, I don't know, this might be life for some of us living in our homes these days. But this is really fascinating to me. So, you know, so the archaea, the methanotrophs, they're, you know, trying to break down the methane, but they're doing it with these bacteria partners that are reducing sulfate at the same time. And, you know, at the end, they spit out some DIC, some hydrogen sulfide, it's really smelly, and then water. Okay, this reaction is so critically important because the largest reservoir of methane on the planet is underneath the seafloor. But 99% of it gets eaten by this consortium of microbes before it ever leaves the seafloor, goes into the water column, and way before, you know, it reaches our atmosphere. So super fascinating. This would be like the #1 metabolism that I would look for if you gave me billions of dollars and, you know, let me go to ocean worlds. Okay. So what's going on here, right? Because it looks like this low temperature equilibration. All right, so now we're going to be looking, you know, from seafloor and below, so about 30 meters depth. Here we are in the Bornholm Basin. I also sailed on IODP 347. This was my first IODP cruise. I loved it because the Baltic Sea is like this classic biogeochemistry site. Like a lot of what we know about the sulfur cycle, the methane cycle, and this anaerobic oxidation of methane, a lot of that original work was done in the Baltic Sea and in Aarhus Bay off the coast of Denmark. So really exciting to get to participate in that. All right, so we can see that both of these proxies we're looking at, 13CH3D and 12CH2D2, They start out at some nominal positive value, and then they increase with depth. Okay, they're heading towards this gray line, that's representative of the black curved line from earlier thermodynamic equilibrium. These sediments were about 7 degrees Celsius plus or minus 2 the whole way down in just this shallow depth here. Had all that methane formed somehow in equilibrium at those temperatures, it would all be on that gray line, but you see it is not. We're sort of moving towards there as we go with depth. Okay, our methane concentrations, something else that's also of interest. We sort of have this like peak, we're like highest right here in the middle, kind of between this white and this blue box, which I'll tell you about in a second. And then there's some sink that, methane's being consumed here in the subsurface. All right, well, we can also look at sort of our traditional proxies, right? For instance, stable isotopes. So we've got the stable isotopes of carbon of methane and of deuterium of methane. And this is where you can see stable isotopes are going to get a little bit tricky. So remember, biology is lazy, right? So we would think, well, where you have a lot of methane, here you would have, a lot of methanogenesis, and you should be producing the lightest methane. But that's not what's shown here in the carbon isotope curve, right? We actually have heavy, relatively heavy methane here, okay? So that's a little bit odd. And then, wow, what's going on in this deuterium curve? Well, so this deuterium is probably coming from water. And I'm gonna tell you what this white and blue box are. During the last glacial maximum, the Bornholm Basin was like a freshwater lake, okay? The Baltic Sea was like cut off from the open marine system. So, freshwater water isotopes are going to be very different from marine, brackish marine. So, which is, what the Baltic Sea has been for most of the Holocene. So, this is sort of that substrate problem here where it's like, oh, hydrogen isotopes are changing, but it could also just be these really different pools of water sort of influencing, this formation of methane over time. Okay, but what's exciting when I see this is that this information here looks different from this information over here. So we have the potential to learn something new with these two new tools in the system where these traditional proxies might be a bit confusing. Okay, well, so what about the other half of our reaction, right? Because we can't just be, making and consuming methane. There's also this sulfur or terminal electron acceptor that's helping us do the anaerobic oxidation of methane. So sulfate is obviously going to be a key one here. So here's our sulfate profile, but particularly in the freshwater sediments, there's a hypothesis that iron oxides might be very important. So we could have sort of an iron dependent AOM type thing. So here's what the iron profile looks like. And then my collaborator, Matthias Egger, sort of did some modeling to figure out rates of sulfate AOM, methanogenesis, and then iron AOM. So you can see, I should tell you also, interesting tidbit. So we don't have any sediments or samples from this part because this part of the Bornholm Basin was likely a World War I ammunition dump. So we were not allowed to collect the upper part of the sediment because it could have had like nerve gas in it. So this will remain a mystery for time eternal, but using basically seawater concentrations of sulfate and iron to sort of reflect what's likely to be in the upper part of that sediment, he was sort of able to model this upper sulfate methane transition zone. So lots of sulfate AOM up here, followed by peak and methanogenesis just below it, and then potentially we have this peak in iron dependent AOM, but then there's also this deep SMTZ. So it's not shown here, but there's also likely to be another peak down here for sulfate dependent AOM going on. Okay, so what's it all mean? Oh, and I will go through this quickly, but there's also lots of molecular biology evidence that shows we have these AOM as well as methanogen critters in this sediment. All right, and so for that, we're going to go back to another one of these, metabolic pathway plots, okay? So it looks a lot like the others, except this time all of the arrows are running in reverse. AOM is often referred to as reverse methanogenesis because it's basically that same machinery, again, with MCR being this really important enzyme, running backwards. So we're starting with methane, and then at the end, we're going to kick out DIC and water and hydrogen sulfide. But it's really all the same stuff, taking off these hydrogens one by one. All right, so what happens here? Think about it this way. This metabolism, I mean, it's so difficult. They actually, they need a buddy to help them do it. So like, just imagine you get home at the end of the day, you're exhausted and you have no energy to eat dinner or very little energy to eat dinner. You know, you take it all in and you're just so tired it falls out of your mouth. Okay, it's kind of a silly metaphor, but that's sort of how I think about this, right? So, you know, these methanotrophs, they're getting that methane in their system, and it's hanging out on this enzyme, right, which has all these metal surfaces. And so we could potentially equilibrate some hydrogen isotopes, but they don't have enough energy to sort of like do that first step where they cleave it off, or rather they don't have it all of the time depending on what their bacterial partners are doing. Okay, but that's a mechanism where you could sort of, you know, get that methane in there, re-equilibrate it, but then just like spit it back out. Say, I don't have the energy to deal with you right now. I'm gonna eat this dinner tomorrow, okay? And so you don't have to believe me, just my, you know, crazy metaphors for this, because there's been some great work from the University of Bremen, Max Planck Institute of Marine Biogeochemistry, that actually shows in closed system experiments that when you have methane that you're eating, so here methane going down, and you know, and with that sulfate that's being consumed by the bacterial partners, you can actually, as the concentrations of these things go down, you can start to sort of equilibrate between the methane pool and the DIC pool. And this begins to happen when sulfate reaches this critically low concentration, about less than 0.5 millimolar. Okay, so keep that in mind. Because when we have lots of sulfate, for instance, maybe we have marine concentrations of sulfate, that back reaction only 3 to 7%. So, you know, it's nominal, but still there. When the sulfate concentrations get low, below that critical threshold, we could have up to 78% back reaction. So think about it, you know, almost as much methane as they're consuming, they're actually not able to consume. They put it back out into the environment, but it's had the chance to sort of re-equilibrate on this enzyme here. All right, so if we sort of go back to the big picture here where I've got methane concentration and all this other stuff up top, and then here are isotope proxies on the bottom. You know, the lines of evidence we have is, you know, so we have a carbon isotope reversal at a place where we think there should be, you know, methanogenesis. Oh, so that doesn't make any sense. We have very low sulfate concentrations in this area. We're in a place where methanogenesis rates are decreasing and AOM rates are potentially increasing with depth. And then we have this clumped isotope equilibration that is really happening sort of right at the point where there's no longer enough methane for me to measure. So it's definitely, you know, it's decreasing to some extent. And so that leads me to believe that anaerobic methane oxidation is actually a mechanism for re-equilibrating pools of methane over time. I'm not going to show this slide, but I calculated how long it would have taken to get this Bornholm Basin methane, you know, re-equilibrated from something that was formed down here in the microbial array. It's on the order of 10 to the third to 10 to the fourth years, which is very consistent with the age of the sediment. But essentially, I hypothesized based on this evidence that we could take methane made in any part of the diagram, give it to anaerobic methane oxidizers, and under the right conditions, they will equilibrate it with their environmental conditions. Okay, and so, you know, bringing it back to where I told you we were headed, and what sort of the state of this tool now, what's really exciting is, you know, not only do we, you know, have sort of this place on the diagram where we think microbial methane is formed, where we have, you know, these potential abiotic places where methane can form. You know, these processes like thermogenesis and AOM, they're very tightly constrained by their temperatures, right? You know, we haven't really found AOM happening any, you know, other than between 0 and 70 degrees Celsius. We have pretty good ideas of where the gas window is, you know, from about 100 degrees Celsius to, you know, something over 200 degrees Celsius. And so these are all distinguishable from one another. And it's also exciting because, you know, AOM is a really important metabolism here on Earth. I think if we start looking at methane cycles in other places and we find life there, it's also going to be very important here. And AOM, when we look at it this way, it's not just a thing that's going to happen to, you know, the methane that we've made some other way. It itself is actually sort of, you know, like a source, a point source on a diagram like this. So that's sort of like a new and different way of thinking of AOM rather than just like, oh, we're going to take methane that was made here and just, you know, alter it in some way. So that's very exciting for me. And we could even potentially, with some further work, sort of use a diagram like this to actually break out and learn something about the biochemistry of specific, metabolic pathways, even within the methanogens. So that's like a super positive place to end, but I will not end there. Instead, I'm going to end here with this mess, because it behooves me to say that, having looked at a number of these samples, of which I only showed you, some of today from environmental systems, that natural samples rarely look like N numbers, right? I mean, it's when you see it all there, you're like, wow, how are we ever going to sort this out? But I think, these tools that we're building in terms of understanding both the physical processes that can occur, to a pool of methane. I had a great conversation with Peter Flemings and Steve and Kawe today about, how, different processes could sort of move things from one diagram to the other, and then sort of keep in mind like the chemistry that actually creates methane and what that's going to look like in terms of building a molecule isotope by isotope. I think we can use tools to help us really, interpret and pull some of this apart. So stay tuned. I have a lot of methane that I'm working on. I'm waiting for the all clear to fly so I can head to UCLA and keep moving ahead with this. But I'm sure it'll happen eventually. And thank you so much. I hope we can all have coffee and cookies together soon, negotiations now. And I'm happy to take any questions.
That was really awesome, Jeanine. Thank you. Yeah, we have plenty of time for questions if anybody has any.
Yeah, Jeanine, I had a question.
Yeah, Steve.
This is Steve Phillips. Yeah, so it's interesting you showed that the isotopologue distribution didn't seem to be affected by reaction rate during methanogenesis. So you think we ever get methane at equilibrium from methanogenesis, or do we need AOM to get close to that line?
Yeah, I mean, so this is really tricky. So I have never seen environmental methane that I think is made, that is not made by any other way, abiotic or thermogenic, that doesn't look like that low temperature equilibrated methane. That's not true of everything, but in the marine system, freshwater lakes, we definitely get some of these like huge kinetic signals in places where I think it's all methylotropic methanogenesis. I don't know, this is sort of like one of those philosophical questions, because there's actually a lot of debate about whether or not anaerobic methanotrophs are also methanogens. So, if you look at some of this work coming out of like Karen Lloyd's group at UT Knoxville, White Oak Bayou stuff, they think they have evidence for methanotrophs that switch their metabolism to making methane and are able to get energy out of it, right? So there's no question in my mind whether or not methanotrophs spit methane out. Are they getting energy from it? Is like a different question. It feels very like, I don't know, Alice in Wonderland. How is the methanogen the methanotroph, and vice versa? But I pity the PhD student that's going to walk into a project that's going to say, okay, well, how low of a rate can you get methanogens to grow? Because then they're looking at trying to incubate things for months to years at a time. And I'm trying to write a paper on the timescale of a PhD about that. So no, this is a criticism I get all the time. And I say, well, I look forward to your decades-long incubation of methanogens growing at really slow rates to answer this question, because I don't know. We really have to couple, I think, with, I'm not a biologist. We have to couple, I think, with closely with microbiologists, and use all of the tools at our disposal to answer questions like this. So probably not satisfying, but that's what I have to say.
Thanks.
Well, I've got a question. So just thinking about toward the end of your talk, one of the things you showed was this role of abiotic metabolism kind of reworking everything to make it look the same. So I guess I'm wondering if we fast forward to, one of the early slides you showed was this sort of drive to understand these systems as a way of fingerprinting, say for looking at Europa. So assuming you could cram your instrument onto a probe or miniaturize it, right? So that or maybe send samples back and wait 20 years to get them back or something. I mean, what are the prospects there? What is the implication? Is there a concern about homogenization of fingerprints so you won't be able to see any of the things you want with this technique for these kinds of processes for those applications?
Yeah, so I think that's a really interesting question. And I think about it a lot. It would definitely be a sample return thing. We're not going to put one of these mass specs on a payload.
Serious payload.
Yeah. So I have hope because, I glossed over a lot of stuff today. But so for instance, when I look at methane in the Weymouth Basin, I do not see signals that have been completely blurred out by, putative anaerobic methane oxidation, right? That's a really complex place where we have these hot, sills interacting with organic rich matter. There's tons of microbial activity in the organic rich matter. What's happening in the hot sills that are like cooled down to, 90 to 100 degrees Celsius? Who knows? We're still working on it, but we get very different populations of methane that they don't look just like AOM. And I showed sort of in this big diagram here, there's a huge range of signals that we get, and these are all natural samples. Okay, these are not experiments, so they're not all going to look like AOM. But I will answer your question with a question, which is, what do you think we would learn if we were just to measure, for instance, the stable isotope composition, considering that we know nothing about the isotopic composition of the substrates available in a place like Europa or Enceladus, right? As much as we know about the composition of substrates on Earth, there are still places where it's not possible to interpret stable isotope signals in an unambiguous way. We'll be going to systems where we literally know nothing about, what would be the isotope composition of, gases coming out of plumes on Enceladus. It's definitely going to be different from what's happening here on Earth. How different? And this is, I said this earlier in my meeting with others, but the dream of every proxy is like, oh, this is going to be the one that we're just going to be able to use this and leave everything behind. That's never the case, right? We all go through a honeymoon period and then we find out how complicated the thing we're working in is. There's nothing different about this system, but it does give us different information than any of these other proxies give us that I hope sort of leads us to a more complete picture, even if we still have a lot of questions at the end.
Any other questions? I was going to ask the same thing. Like, how would we even know if we went to Europa or whatever, what the baseline conditions were that you'd even have? Because especially like you think about like solar system processes, like how do you know that those planets didn't get a whole bunch of the, overwintering in Antarctica isotopologues?
Yeah.
Right. So yeah.
Yeah, no, we're at the lab I did my PhD in. My advisor was primarily a cosmochemist, and so I was working with him on biogeochemistry. And so, we all get sort of stuck in our own little crowds, but every once in a while I like see these plots of, like nitrogen isotopes like throughout the Milky Way. And like the signals are just, thousands of per mil. And you realize like, oh, I know nothing about nothing, right?
Right.
It's going to be the same here. Yeah.
Yeah, we try to use the Earth as an analog for everything else, and Earth is extremely unique, I think is the message that I always take away from these talks as a simple micropaleontologist. But anyway, if nobody else has any questions, thank you all for joining us. And I'd like to remind everybody that we have a happy hour today at 5. You should have a Zoom link. If you don't, send me an email. I'll send it to you. All are welcome. Please join us so we can keep talking about isotopes in the solar system. and mustard gas in the seafloor and everything else. So yeah, Jeanine, thank you again. That was a really cool talk.
Yeah, thanks a lot. It's been a pleasure. I look forward to the rest